Okay, next we will study about the steps in translation. The first step of translation is activation of amino acid and charging of tRNA, otherwise called as amino acylation of tRNA. The second step is initiation of polypeptide synthesis. Third step is elongation of the polypeptide chain, and last step is termination or stoppage of polypeptide chain. Okay, so these are the 4 steps in translation. We will study it one by one. Now, we have already discussed that this translation process happens in case of eukaryotes in the cytoplasm of the cell. The first step is called as the activation of amino acid and charging of tRNA. Now, what is the meaning of it? So, we know that the amino acids are already present in the cytoplasm. When we take in food, the nutrients in the food is absorbed by the blood, and every cell is having all these important nutrients. So, amino acids are also an absorbed nutrient. So, it will be there in the cytoplasm of the cell. Now, but these amino acids are in an inactive resting stage. So, it should be made activated. So, that is done by ATP molecule and magnesium ions. So, in presence of ATP molecules, magnesium ions and the enzyme, amino acyl tRNA is synthesized. What happens? The amino acids will get activated and it will chemically bind to the cognate, means its corresponding type of tRNA. Okay, and that process is called as amino acylation of tRNA. So, what is amino acylation of tRNA? The amino acid binding to the or chemically binding to its cognate or the corresponding tRNA is called as amino acylation of tRNA. So, the amino acid, since it is in an inactive state, it should be made into an active state, and that is done with the help of ATP molecules and magnesium ions. So, first we will learn up to the